Welcome to this explainer. Today, we're doing something absolutely wild. We're decoding the complex laboratory blueprint used to build microscopic medicine factories. We're going to look at a fascinating suite of molecular techniques that let us literally refactor nature, taking the hidden potential of uncooperative organisms and turning them into highly efficient, living manufacturing plants for life-saving medicines. We really have to start by acknowledging that fungi are absolute treasure troves of medical miracles. For thousands of years, we've tapped into their bioactive properties. From the revolutionary discovery of penicillin back in the 1920s to modern immunomodulators like cyclosporin, and even those statins millions of people take every day to lower their cholesterol, fungi have quietly been one of humanity's greatest pharmaceutical providers. But here's where we hit a massive scientific roadblock. What on earth do we do when a fungus holding a potential cure stubbornly refuses to grow in a lab? Many wild fungal species are just incredibly difficult to culture. Or even worse, they produce vanishingly low amounts of these natural compounds under laboratory conditions. We literally know the medicine is locked in their DNA, but we simply can't get them to make it for us. To solve this, here is our master blueprint for today's explainer. We'll start with fungi's hidden medical treasures, move on to meet the surrogate host, and then designing the DNA blueprints. From there, we'll cover assembling plasmids and yeast, booting up the fungal factory, and finally, harvesting the chemical bounty. Let's jump right in. All right, let's kick things off with Section 1, Fungi's Hidden Medical Treasures. So, getting to these hidden treasures all comes down to a crucial process called heterologous expression. Think of it like playing molecular detective. Thanks to modern genome sequencing, we can pinpoint the exact gene clusters inside a stubborn fungus that code for our target drug. Heterologous expression is basically extracting those specific biosynthetic genes and booting them up inside a completely different, much friendlier host strain. We're essentially ripping the factory equipment out of a collapsing building and installing it in a brand new state-of-the-art facility. Which brings us nicely to Section 2, Meet the Surrogate Host. Let's check out our new biological factory floor. We've got a classic problem-solution setup here. On one side, we have our native producer. It's difficult, unyielding, and downright frustrating. On the other side, our solution, Aspergillus oryzae strain NSAR1. Now, Aspergillus oryzae is a domesticated fungus with a massive history in food production, meaning it has a GRAS, or generally recognized as safe, status. It barely produces any of its own natural products in the lab, which is actually perfect for us. It gives us a blank canvas. Better yet, this specific NSAR1 strain has been engineered to be quadruply auxotrophic. That means it's missing 4 specific genes: NEA-D, SC, ARGB, and ADA. Because it desperately needs those genes to survive, we can use them as selectable markers to force the fungus to accept up to 4 separate sets of our engineered DNA all at once. Taking a bird's-eye view of this entire construction project, you can trace the path from the raw biosynthetic gene cluster all the way to the end. We use yeast recombination to build our expression vectors, transform them into our Aspergillus oryzae protoplasts, and finally, we end up screening for bioactivity and purifying the chemical compound. It is literally a masterpiece of biological engineering. But you can't build a factory without precise instructions, right? So let's move to Section 3. Designing the DNA blueprints. Our master blueprint here is the pTYGS expression vector. It's a circular piece of DNA intricately designed to safely house and, more importantly, loudly express the genes we want. We use 4 separate expression cassettes, specifically the AMEB, GPDA, ENO, and ADH promoters. Think of these as massive biological megaphones that force our fungal factory to manufacture the inserted genes at maximum volume. And since we have 4 cassettes per vector and 4 possible selectable markers, we could theoretically coordinate the expression of up to 16 different genes simultaneously. That's huge. Now, depending on the sheer size and complexity of the genetic machinery we're building, we've got options. We can use highly precise yeast recombination strategies to patch our desired genes straight into the vector's expression cassettes. We could insert genes into all 4 cassettes, maybe just 2, or even swap out some cassettes entirely for larger gene sequences. It's an incredibly modular, plug-and-play system. Blueprints drafted? Check! Let's head to the assembly line in Section 4, Assembling Plasmids in Yeast. Okay. We're not just randomly mixing liquids here. We are leveraging a living organism to do our microscopic welding. By using a very specific lithium acetate and PEG method, combined with a precise 42-degree heat shock, we essentially force Saccharomyces cerevisiae, common baker's yeast, to act as an assembly line. The yeast naturally performs homologous recombination, flawlessly stitching our overlapping DNA fragments together into a complete circular plasmid without us having to use messy traditional ligation enzymes. Once the yeast does its job, we kick off an awesome microscopic relay race. We grab that completed blueprint from the yeast and hand it off to E. coli, which acts as our biological photocopier. E. coli rapidly amplifies the correct blueprints, so we end up with millions of copies. And to guarantee it only copies the successfully built plasmids, the original empty vector contains a toxic gene called CCDB. If the vector wasn't properly overwritten with our new medicine-producing genes, that toxic gene stays active and wipes out the incorrect copies. It's a brilliantly ruthless quality control system. So, with millions of perfectly assembled blueprint copies in hand, let's move to Section 5, booting up the fungal factory. To actually get our blueprints onto the factory floor, we've got to bust through the fungal cell wall. We do this by germinating the spores and hitting them with highly specific lysing enzymes that literally digest their tough outer layer. This leaves us with naked, super vulnerable fungal cells called protoplasts. We then mix these protoplasts with our amplified plasmid DNA and a PEG solution, and without their rigid walls, they essentially just swallow our new genetic code. After plating them on a nutrient-rich agar, they rebuild their walls and boot up as fully functional, genetically upgraded factories. When you pull back and look at the whole journey of this DNA, it's wild. It originates from a stubborn, uncooperative fungus. Then, yeast steps in as the welder to assemble it. E. coli tags in as the photocopier to amplify it. And finally, Aspergillus oryzae boots it up as the master manufacturing plant. We're leveraging the unique superpowers of 3 completely different microscopic species just to make one fungus do what we want. The factory is officially online and running, so let's hit our final stop, Section 6, harvesting the chemical bounty. To get the actual medicinal product out, we have to scale up. We grow our transformed fungus in a liquid broth for about a week. Then, and this is great, we literally blend the fungal mycelium up with a hand blender. We break those biological factories wide open. Next is a rapid-fire chemical extraction. We hit it with heavy solvents like ethyl acetate, acidify it with hydrochloric acid to pull out the organics, vacuum filter the debris, dry it out, and concentrate it all down in a rotary evaporator. After all that industrial scale-up, we're left with a highly concentrated, purified chemical extract. Finally, the moment of truth. We have to see what we actually made. We take that extract, resuspend it in high-grade acetonitrile, and run it through high-tech chromatography, like HPLC mass spectrometry. This lets us look at the exact chemical peaks and molecular weights of our output. We can see precisely what new, potentially life-saving molecules our engineered factory built completely from scratch. Which brings us to the ultimate takeaway from this explainer. Knowing that we can bypass the stubbornness of nature, that we can literally draft, assemble, and boot up living pharmaceutical factories to pump out incredibly complex molecules, what untapped cures do you think are just waiting to be discovered next? The blueprint is in our hands. We just have to decide what to build. Thanks for hanging out with me for this deep dive into heterologous expression. I'll catch you in the next one.